And the next speaker is Shubham Chandak, and he's from Stanford University and will talk about large block-length LDPC codes for Illumina sequencing-based DNA storage.
Thank you. Hi. So I will speak about error-correcting codes for DNA-based data storage. So first of all, just to make things clear, DNA-based data storage means storing data in DNA. This is not data compression where we store genomic data on our usual computers. And we will see why we are doing this. So we'll start with the motivation and the setup, because I think this is a relatively new field and not familiar to many of you. Then I will talk a bit about what we did for both Illumina sequencing-based DNA storage and Nanopore sequencing-based, and we will end with conclusions. So as you might have seen before, like the amount of stored data is growing exponentially. Right now, video data is the biggest contributor, but very soon, genomic data, astronomical data, virtual reality, all these things will grow very fast. And right now, the main storage medium is hard drives, solid-state drives, tape, which have some limitations in terms of the storage density, that is how much data you can store per gram, and also in terms of the durability. So recently, people have started looking at other alternative storage media. So let's say you want to store 200 petabytes of data. So if you use hard drives, you will need like a truckload of hard drives, and it will last for a few decades. On the other hand, if you use DNA, it will fit in this small 1 gram tube. It can last for thousands of years in the correct conditions. And plus, DNA as a storage medium has a lot of advantages, such as easy duplication, which our cells do all the time. So this is a recent news where a startup, they were able to store the entire Wikipedia text, which was around 16 gigabytes, in this DNA, which you see. And obviously, this is like very small, 16 GB you can store in a pen drive. But yeah, so this is an emerging thing, and very soon we might be able to store petabytes of data. So let's talk about how we can store data in DNA. Obviously, like the nature does it all the time, but let's see how we can store some file in DNA. So first of all, right now we have ability to synthesize short DNA fragments, artificial DNA, single-strand DNA, of length around 150 nucleotides. And this can be done at scale. You can synthesize millions of these sequences in parallel. Right, now let's say you want to store a binary file on your computer. So the obvious thing is, okay, you segment the binary file into smaller portions because you can only have 150 length sequences. And then you convert this binary into DNA using some standard encoding. Of course, once you put the DNA in solution, you lose the ordering of the sequences. So you need some sort of indexing so that after you read it back, you need to be able to figure out like which sequence was at which position. So you add some indexing. Then again, when you read back the DNA, you don't expect to read back each and every sequence you put in the solution, right? So some of the sequences will have zero coverage just by the randomness. And therefore, you need some sort of error correction or erasure correction. And this is done all the time in traditional storage systems. So what you do is, in traditional storage system, you have some K data disks, and you basically add some extra parity disks, which store some redundant data. And later on, let's say you lose 3 of the disks, you're still able to get back everything. So you can do basically the same thing for DNA storage. Finally, as we all know, both the synthesis and the sequencing processes, they cause a lot of errors, substitutions, insertions, deletions. And therefore, we also need to add some extra parity or some redundancy so that if there is some error, like this one, you are able to correct it. Right, and of course, error correction has been studied very extensively in communication and in traditional data storage in the fields of information theory and coding theory. And we are basically applying that knowledge to DNA storage with its unique challenges. So in summary, error correction or erasure correction enables us to store data reliably, even when the synthesis and sequencing processes are noisy and low cost, right? And so I would like to point out a difference from standard biological analysis, which we do, where accuracy is very important, and here, accuracy of sequencing or synthesis is not that important because we have the ability to design our sequences. We decide what we want to synthesize. It's not as if nature has decided that this is the DNA and we want to read it as accurately as possible, right? So since we have the freedom to design things, we can actually handle much more noise, and therefore to reduce the cost, it's actually better to have a low cost noisy system rather than a very high accuracy system, which is very expensive. So let's summarize this in this. So we start with a binary file, we segment things, then we apply error correction codes. We will come back to this later. Then we synthesize the DNA, then we store it for some time. So right now, most people are thinking of DNA storage in the for archival purposes, but we never know. Finally, when you want to read the data back, you do sequencing, maybe Illumina, maybe Nanopore, maybe something else, and you do the base calling process to get back these sequenced reads. And here you have lots of errors, as we already talked about. Some of the sequences you see twice, the ordering is lost, some of the sequences are lost, and even the sequences you get are corrupted. And then using the error correction codes, you should be able to decode what you actually put in the solution. Right, so we look at two sequencing platforms for our project, Illumina sequencing and Nanopore sequencing. So Illumina is good in terms of throughput and also in terms of very low error rates. Especially, it has substitution errors, which have been more widely studied in the communication literature. On the other hand, Nanopore has some advantages such as portability. It's very small. It's real time. It can produce long reads. But the error rate is very high, and it has also insertions and deletion errors. So both of them have different challenges, and we will see how we try to solve them. So there has been a lot of work in the past 10 years on DNA storage, and people have looked. So it started with a paper by Church and others from Harvard, but after that, people have done a lot of work on aspects such as error correction codes. People have looked at, let's say I want only one file out of my, from the DNA, I don't want to read the whole thing. So we can still do that using PCR. People are looking at scalable and cost-effective synthesis, different sequencing platforms, theoretical analysis. So, but there are some limitations. In particular, many of the works, even though they do error correction coding, they rely a lot on consensus. So they do a really high coverage reading, and then they collect the reads from the same sequence, and then they do some sort of consensus, which is really inefficient in terms of the coverage you need. So we try to solve that in this work. Right, so what is our contribution? So first of all, we study two fundamental quantities in a DNA storage system. One is the writing cost, which is to store one message bit, how many bases are you synthesizing? And the other is the reading cost. To decode one message bit, how many bases are you sequencing? And note that the reading cost is a bit different from coverage. This is a subtle point, but most of the previous works used coverage, which is not really the right metric here. Coverage is actually bases sequenced per basis synthesized, which does not make much sense here. So in our work, we study the theoretical trade-off. There is a trade-off between the writing cost and the reading cost. Basically, if you do more error correction, then writing cost becomes high, but the reading cost becomes low. We develop schemes which can achieve better trade-off by reducing the reliance on high coverage. This I will come to later. And finally, for Nanopore, we did something really interesting to be able to exploit the information in the raw signal rather than looking only at the base called sequence, which has very high error rates. So we will look at all these. So let's go to the Illumina sequencing-based DNA storage. So what's the key idea? Basically, most of the previous works, what they do is they start with the data, they segment the data, and then they do two separate levels of error correction. One is for the loss of sequences, and the other is for the errors within the sequences. What we do is we just apply a single code, And the problem with this is, like, you can show in information theory that if you have short sequences, there is a fundamental limit on how good error correction you can do. On the other hand, if you do it on a large block, it's much better. And so we do that. And plus we use like a lot of other heuristics for handling deletion errors, insertions, etc. So let's see. So what we did was we did like some small experiments storing around 200 kilobytes of data. We did synthesis of length 150, sequencing with Illumina. And like the interesting thing is that even though Illumina mostly causes the substitution errors, 0.4%, there are lots of deletions and lots of deletions due to the synthesis. And plus, note that our synthesis technology we used was cheaper, but also noisier as compared to the previous works. This will be important when we see the results. Right, so here I show the results. So we see the reading cost on the x-axis, the writing cost on the y-axis. This direction is better, like you want to go towards this direction. So we compare to two previous works. One is by Microsoft Research and University of Washington. The other is from Columbia. And we see that in general, we are on the good direction. Like at the same writing cost, we achieve better reading cost in general. Of course, because of the variability in the experiments, plus because our synthesis was a bit more noisier than the other works, our results are, like if it's not an apples to apples comparison, like I think we can be even better if we use the exact same synthesis and sequencing. Now let's look at what we did for Nanopore. So Nanopore has unique challenges, right? Because you have error rates 10 to 15%, and it's very hard to develop error correction for these. So most previous works just do very high coverage, like they do 30x, 60x, and then they take like multiple sequence alignment or consensus. So we thought, why not do something new? So we started looking at the Nanopore as a communication channel, as a model. So basically the DNA goes in, and there is a pore, the DNA goes through the pore, and what you get out is this raw current signal. And it's a very bad channel from a communication point of view. There are all sorts of non-idealities which you don't want. Right, so it's very hard to model and analyze and like actually develop codes for this. So we thought, why not use machine learning, which can help us do the modeling part, and then use the coding, error correcting codes on top of that. So let's see what we did. So we started with the Flappy base caller, which is the open source version of the Guppy base caller by Nanopore, which is the official Nanopore base caller. So what it does is it starts with the raw signal. It applies a bunch of neural network layers, and it gives out some probabilities, which are some sort of transition probabilities. Now, what happens usually is we do the base calling from these probabilities. And since the base caller has no idea about the error correction code we applied, so it is like basically blind to the error correction code. So oftentimes it makes an error. But what if we do, instead of like going this route, we do decoding, where we use the constraints of the code. So basically, instead of just doing base calling, which is blind to the code, we do some sort of decoding process where we use the constraints imposed by the code. And here it's to be expected that the success probability will be much higher. And to get a slightly better sense of what goes on here, like basically the base caller has some sort of Markov model, some sort of hidden Markov model where they do some transitions from, like at this time, base A is in the pore, at the next time base C in the pore, so on. And the error correction code we use also has a very similar structure. We use a convolutional code. And what we do is, in the decoding, we just combine these two to get a big like trellis, and we do like standard Viterbi decoding for the hidden Markov models. So some preliminary results for the Nanopore approach. What we see is at similar writing costs, we see like 3 to 6 times lower reading costs. Just to compare, like for Illumina, we had like 30 to 40% better results, but for Nanopore, the advantage is huge because we are using the raw signal. In particular, many of the sequences, just by looking at one copy of the sequence, we are able to decode successfully, which is basically impossible if you directly use the base called sequence. So using the raw signal gives you like much more power. And yeah, so this suggests that the raw signal has much more information than base call sequences. And like, of course, people are also using this idea in other bioinformatic applications. Like you might have heard of nanopolish, which improves the assembly using the raw signal. Right, so in conclusion, we have introduced novel coding schemes for both Illumina and Nanopore-based storage systems. The Illumina scheme is under review, and it will be up on bioRxiv next month. The Nanopore scheme we are writing up right now. And of course, like a DNA storage system has a lot of aspects. It's not just about the error correction. You also need some random access capabilities. You need an ability to read the same data again and again. So we are working with collaborators to implement those things as well. In the long term, we envision going towards nanopore sequencing since it's portable and combine it with like cheaper and noisier synthesis techniques. And the good thing about the base caller decoder integration idea is that even if you have like some other type of synthesis, right now you can do base by base synthesis. You give a base sequence, they give you back the sequence. Maybe you can do like k-mer by k-mer synthesis, something else. This strategy still works. Right, and of course, like the core idea is applicable beyond DNA storage. So in bioinformatics, like rather than just working with the raw signal, how about we work with the, just working with the base called sequence, why not work with the raw data? And similarly for communication. Right, so it's a big project with people from both electrical engineering and from the bio side. And we are funding from NSF, and so I would like to thank everyone here. Thank you. Please come to the poster session if you like.
Thank you very much. So we have time for a few questions.
Hello, thank you for your talk. I just wanted to ask if you could talk a little bit about dealing with the insertions and deletions directly in your error correction schemes, in particular the Illumina scheme, where you don't have inner and outer codes, you just have this one long code. How do you deal with insertions and deletions?
So let's see. I think I have a few slides, right? Yeah, so basically what we do is, so one new thing which we did was like in everything we add this small synchronization thing, AGT. So what happens is when you read things back, let's say like in your sequence, there was a deletion here. So note that the deletion rate is really low. You would expect maybe 1 deletion per sequence. So maybe you have a deletion here. So looking at this marker, you can guess, okay, this part was deleted. There is a deletion somewhere, right? And the main thing is like the LDPC code can handle erasures. So this large block code can handle both erasures and errors. So if there is a deletion, if you can like throw away the sequence, that's also fine. And when we do some like multiple sequence alignment, so if I have a bunch of reads from the same sequence, the multiple sequence alignment can find out the deletions. So there are more systematic approaches which we can do, like so that's part of future work.
Cool, thank you.
Hi, so thanks for your talk. I was really intrigued by the nanopore base calling and decoding at the same time. Can you elaborate a little bit more on that? Because I could not follow it exactly.
Yeah, so basically what happens in the base caller is you get these probabilities after the neural networks. And the probabilities are basically at every time step, you have a state which is like the current base, and you can either go to a new base or stay at the same base. So like because the nanopore, like you get multiple samples at the same position when the DNA goes through the pore, right? And what the base caller usually does is it tries to find the best path. So this is like a hidden Markov model thing. So you try to find the best possible sequence of transitions which give you this particular raw signal, something like that. So that's the standard base calling process. What we do is we basically disallow any paths that are not valid code words. Because we are applying an error correction code, so we are restricting the space of possible sequences.
So how do you know what is valid?
Oh, that's because that's due to our code, right? Because since we have this convolutional code, so like in error correction codes, because you have some amount of data and you add some parity. So the parity is determined by the data. So if you have some arbitrary binary string, that might not be a valid code word.
Then you need to read first the parity bits.
Yes, actually the code is a bit more complicated. Like it doesn't really separate the parity. Parity is sort of mixed in the whole thing. So it's slightly more complicated, but the idea is, Like the idea is that the convolutional code we use, the error correction code, it has a very, very similar structure to the nanopore base calling process. So they like fit together very nicely, and you can easily restrict yourself to only the sequences that are valid code words.
But don't you have the same problem in reading the parity bits then?
Yeah, yes, as I said, like yeah, maybe we can talk offline. Like the parity bits are sort of intermixed in the sequence. It's not like they're separately stored. So it's a bit more nuanced.
Let's talk afterwards.
Yeah, yeah.
You can have one last question.
I was just wondering how changes at Nanopore are going to affect your ability to read and write here. So if Nanopore changes their default strategy to the 1D squared approach, how is that going to affect anything that you've already built?
Yes, I think for 1D square, I think the error rates go really low. So then maybe we don't need this much work. We can just work with the like standard Illumina codes. Because for 1D square, I think the error rates go below 1%. Like there, that's very good. But
Yeah, but I guess I'm concerned about the archival strategy is if you plan to do it using the current chemistry and then they by default only do the 1D squared in 10 years, if they're still around, then are you able to still get back what you put in?
yeah, I think so, yeah. Because the decoding you can modify according to the particular technology. Because as
So
long as
you
know...
can change your decoder.
Exactly,
basically.
yeah.
Ah,
Thank the speaker again. And the last speaker of the session